I think someone's background noise is a bit louder.
Sorry, was that me? I was just moving some pens, but I did hear something else on someone else's earlier.
Okay, so yeah, you can unmute yourself so you can later on, ask questions. So the dietary carbohydrate consists of starch, disaccharides, and monosaccharides. So disaccharides and monosaccharides are the simple carbohydrates, and starch is a complex carbohydrate. Monosaccharides does not require any digestion. The polysaccharides and disaccharides needs the digestion, so which means it has to be converted to monosaccharide. Both your disaccharides and starch polysaccharides have to be converted to monosaccharide. Only monosaccharide can be absorbed. So one of the questions we always ask, why digestion is important? Because only monosaccharides we can absorb, disaccharides we can't, and polysaccharides we can't. Okay, so that's why digestion is important. So where does the digestion begin? In the mouth? Yes. Nothing happens in the stomach and mainly in the small intestine, right? So what is the enzyme that is produced in the mouth that act on starch?
Amylase?
Saliva.
Yes, you are right, amylase. What does it do to starch?
It breaks.
The alpha 1,4 glycosidic bond. Okay. So what do you get when amylase acts on starch? The product.
Monosaccharides.
No, you won't get monosaccharides. You get some maltose, okay, and small polysaccharide, which is known as dextrin. So complete digestion doesn't happen, okay. Partial digestion happens in the mouth, okay. So these are the product that you get. Amylase acts on alpha 1,4 glycosidic bond, producing some maltose and dextrin, small polysaccharide, okay. Now, nothing happens in the stomach. Why? Stomach?
There's no enzymes there to
No
break it down.
enzyme there which can act on carbohydrates. Stomach. So lack of enzyme. And the pH, right? Highly acidic pH. So whatever amylase has gone into the stomach will be inactivated by the hydrochloric acid. So no carbohydrate digestion happens in the stomach. Then in the small intestine. Okay, so here we have which enzyme? Pancreatic amylase, correct? Which comes from the pancreas. So what the pancreatic amylase does is it acts on the small polysaccharide, which is known as the dextrin. So it acts on that. So what do you get from there? Maltose. Okay. Now we need to break down all the disaccharides either coming from the diet or the maltose which you get from starch. Okay. So what are the other disaccharides? Lactose and sucrose. So these will be acted upon by enzymes produced by the intestinal mucosal cells. So they are known as the disaccharidase. So here we have maltase, lactase, and sucrase. Okay. So they are grouped as disaccharidase enzyme because they act on disaccharides. Okay, maltase, lactase, and sucrase. So all these disaccharides are going to become, maltose becomes glucose plus glucose, correct?
Yeah.
Lactose is going to become gal plus
Galactose.
glucose. Yes. Sucrose becomes
Fructose plus glucose.
correct. Now we have all the monosaccharides. Okay. Only monosaccharides can be absorbed. So this is done by active transport. Okay. Active transport. Okay. Means ATP is needed. Sodium dependent. Okay. Co-transport, which also uses actually ATP. So this happens in the brush border epithelial cells. Okay. And all the monosaccharides are going to go through that. So from there into the blood. Okay. So into the blood. From the epithelial cells, intestinal epithelial cells into the blood. From the blood to the cell. It can be liver or it can be any cell. Okay. It can't get into the cell. So when glucose is high in the blood, right? After a high carbohydrate meal, blood glucose is going to go high. So that is the signal for release of a hormone from the pancreas, the islet cell, which is the hormone?
Insulin.
Insulin. So insulin is released in response to high blood glucose level. So what insulin does is, through a second messenger system, recruits receptors, glucose receptors. GLUT1, GLUT2, GLUT3, there are different types of receptors. So don't worry too much about that. GLUT receptors are activated and recruited onto the cell surface. Yes, GLUT receptors activated, and once it is recruited and occupies the cell surface through which glucose goes in. Okay. So this is by facilitated diffusion and using glucose receptors or GLUT receptors. Okay. Are you okay with that? This is what we call uptake. Uptake of glucose into the cell. Okay. Uptake.
Can I just check the GLUT receptors? They're attached to they're on the cell.
Surface of the cell.
Okay. Yeah.
Through which glucose is going to go. Because
Yeah.
glucose is highly polar. It can't go through the phospholipid bilayer. That's why you need a receptor.
Okay.
And this receptor is specific for glucose. Okay.
Yeah. And it's the insulin that activates the glucose receptor.
Correct.
Okay.
Yes. Insulin also does so many other things. Insulin activates the phosphatase enzyme, insulin bind to the receptor known as the insulin receptor. So then it gets autophosphorylated, and then it activates the phosphatase enzyme. When phosphatase enzyme is activated, it is going to activate some of the steps in glycolysis, some steps in glycogenesis, some steps in lipogenesis. That's all effect of insulin. Okay. So don't be under the impression that insulin is going to carry glucose into the cell. So that's the way we actually put it in the slides, correct? Yes. Insulin takes glucose into the cell, but it is not, insulin is actually binding to the glucose and taking into the cell. Okay.
Yeah.
Are you all right with that? So many times I find students writing like that: insulin, takes glucose into the cell, but insulin is not going to take it like that. Okay. Are you clear with that?
Yeah. Thanks.
Yes. When you write an assignment, you need to be a bit more clear when you write that, otherwise you might lose mark. Okay.
So insulin binds to glucose into the cell. No,
Insulin can't do that. See, insulin binds to a receptor which is found on the cell surface. It's known as insulin receptor.
Okay.
So that receptor gets activated, and then through second messenger system, it recruits glucose receptors, GLUT receptors, through which glucose gets into the cell. Okay.
Yeah.
All right. So once glucose is inside the cell, what happens to this glucose? Glucose goes into glycolysis. Okay. Insulin activates this process. So there is an enzyme, a regulatory enzyme in glycolysis, which is step 3, fructose 1,6 biphosphatase enzyme, phosphofructokinase, sorry. Step 3, PFK, phosphofructokinase enzyme. So this enzyme is activated by insulin, step 3. So that's why glycolysis is happening. So it upregulates or enhances glycolysis by a 10-step reaction. So you are going to get 2 pyruvate, 2 ATP, and 2 NADH. So that's the output. The input is going to be that. That's the input, and this is the output. And the important reactions are 1, 3 uses ATP used. So they are kinase enzyme. Step 6 produces NADH. And step 7 and 10 produces ATP. So 2 ATP is used and 4 ATP produced. That's how you end up with a net of 2 ATP. So this importance of glycolysis is it's a preparatory process for producing more ATP from glucose. When we have produced enough ATP, glycolysis is going to slow down. Okay? Because the step 1 is where glucose becomes glucose 6-phosphate. When you have more glucose 6-phosphate, hexokinase is going to be inhibited. So glucose 6-phosphate is going to glucose to glucose 6-phosphate. That is your step 1. This becomes glucose 1-phosphate. Yes. And then that becomes glycogen. That process is known as glycogenesis. Okay. So insulin also activates glycogenesis. Insulin activates glycolysis. Insulin activates glycogenesis. Okay. Now, any questions so far? Where pyruvate can go? Pyruvate under aerobic conditions become acetyl-CoA. So this also needs NAD plus becomes NADH. Yes. And it's known as oxidative decarboxylation. So a carbon dioxide is lost there. Pyruvate, and this also needs coenzyme A. Okay. So it's a multi-enzyme complex. The name of the enzyme is pyruvate dehydrogenase complex. Okay, it's a complex enzyme, multi-enzyme. Requires NAD, coenzyme A as coenzymes. And NAD becomes NADH. So you can see a 2-carbon compound is produced. This is a 3-carbon and that's a 2-carbon compound. Okay. This happens in the mitochondrial matrix because that's where the enzyme is present. And it happens in presence of oxygen. If oxygen is not present, then that becomes lactate. Yes, that is anaerobic metabolism. So this is going to use NADH, NAD plus will be produced, correct? And that NAD plus is going to be used for step 6 of glycolysis. Lactate will be carried by blood to the liver. Liver converts lactate to pyruvate. Yes, and pyruvate then by a series of reactions becomes glucose. that is gluconeogenesis. This is what we call the Core's cycle. Remember Core's cycle? Formation of lactate and utilization of lactate. Okay. Pyruvate also can become ethanol. This happens in yeast. Yes. Pyruvate becoming ethanol. Pyruvate first becomes acetaldehyde and then to ethanol. All right. So that's the metabolic fate of pyruvate under different conditions. Are you all right with that? Okay, guys. So now what I will do is, let's see how we have done with our teach. I will share my screen. Not that one, sorry. One minute. All right, are you able to see this one?
Yes.
Yes. So now what you do is you just type in kahoot.it and then this pin you need to put in. Are you able to get in?
Yes, I'm in. Thanks.
Two of them are there. So what about the other two? Yeah, three. One more. All right. I think we are on only 4 people at the moment. Okay. That's all right. Okay. So this is on carbohydrate metabolism. Okay. So one, the digestion of carbohydrate starch in the mouth requires which enzyme? Is it lipase, amylase, protease, and nuclease? Well done. Okay, so that is the correct answer, amylase. So we'll go to the next one. True or false questions. Importance of digestion is only monosaccharides can be absorbed. There is one choice which is wrong, right? False, somebody chose, because you can't actually absorb disaccharides or polysaccharides. Yeah, only monosaccharide can be absorbed. So that's the significance of digestion. Okay, so you need to say somebody is yeah, that's all right. So that's how we know which concept we have to go back and look at, right? All right. Next. Now, the uptake of glucose into the cell requires. So a hormone is needed. Is it glucagon? Is it insulin or inulin or somatostatin? Glucagon is not the one which is needed. Glucagon is produced when there is low blood glucose level. Glucagon helps in making glucose inside the cell and then allows the glucose to go out of the cell. Yes? So that's the role of glucagon. Insulin and glucagon, they are the counter-regulatory hormones. Insulin is produced when there is high blood glucose level, and when insulin is released, glucose is going to be taken into the cell through this second messenger system. As I said earlier, glucagon is produced when the blood glucose is below the normal. So the normal reference range is 3.5 to 5.5 millimoles per liter. So when it is below 3.5, that's when glucagon is going to be released, so that you can increase the glucose level, bring it back to that range. Yes? Are you clear with that? Hello?
But it says that it's inulin. Is that not the right answer?
Inulin is a carbohydrate. It's not a hormone.
No, what is the answer? It's not. I don't.
Insulin. Oh, sorry. Apologies. It should be insulin. I
put insulin and it's
yeah. Sorry. I need to fix that one. Insulin is the correct one. There is a mistake has gone in there. I'll fix that one.
Oh, that's right. I was looking at it because it said inulin.
No,
I'm like, oh, what have I missed?
You haven't missed. So,
I was like,
so it is my mistake in choosing. I meant insulin, and somehow I don't know. So, okay.
Cool.
Inulin is not a hormone. It is a carbohydrate. Okay.
Okay. Cool.
Yeah, it is actually a polymer, polysaccharide. Sorry for that. Okay, you picked it up. Good. Yes, I will fix that one. Yes, I just in a hurry made it. Okay.
That's all right. Hey.
Input of glycolysis. Excellent. Yes, glucose is the input and output is pyruvate. Okay, so that's all good. Which step in glycolysis uses ATP? 1 and 3, 7 and 10, step 6 or step 2? Yes, step 1 and 3, right? That's the correct one. 1 is where glucose becomes glucose 6-phosphate, and 3 is where fructose 6-phosphate becomes 1,6-biphosphate or bisphosphate. Both steps needs ATP, yes. And this one 7 and 10 produces ATP, yes. And 6 produces NADH. Step 2 is just an isomerization reaction. Are you okay with that?
Yeah.
Okay.
Yeah.
Good. So in your write-up, you don't have to discuss all the steps. Okay. You can say the important steps where ATP is used or produced, and step where NAD is used, which becomes NADH. So that way, you are touching on the important steps of glycolysis. Okay, are you all right with that?
I have a question on the steps.
Yes.
The step 6. So the step 6 doesn't require the ATP to create another phosphate. Is the NAD?
Yes, it requires
plus.
NAD. Step 3 is where NAD plus is used, which becomes NADH. So glyceraldehyde 3-phosphate becomes 1,3-bisphosphoglycerate. Yes, that's the step.
The step 6. The step
Yeah.
6. Where is
it uses a
oxidation?
phosphate. It doesn't use ATP. Instead, it uses an inorganic phosphate.
Okay, the inorganic phosphate. Oh, okay. But it comes from NAD plus, or where does it come from?
No, NAD plus don't have any phosphate. That it picks up from the cytosol. It takes up from the cytosol. Remember, in the cytosol you have inorganic phosphates.
Okay.
Yep. Yes. So that phosphate polyatomic ion is picked up from the cytosol.
Okay.
Glycolysis is happening in the cytosol. Okay.
Yep.
moving on to the next one. True false question. In glycolysis, steps 7 and 10 produces ATP.
True.
Mhm. Good, well done. See, those are the steps that you need to remember. You don't have to write exactly what happens there. In step 7, 1,3 bis-phosphoglycerate becomes 3-phosphoglycerate and so on. So because you don't have so many words to write in your assignment, right?
Yeah.
Okay. Instead, you can say it's what is the significance of those steps. Okay. Now, under aerobic conditions, what is the fate of pyruvate?
I made a big boo-boo.
No, that's okay. That's how we learn, As long as we can correct the mistake, that's good, All right. So it is acetyl coenzyme A, right? And the lactate is found under anaerobic condition. And acyl-CoA. Acyl-CoA is nothing but fatty acid with a coenzyme A. Okay. Fatty acid is activated, which requires coenzyme and ATP. That is your acyl coenzyme. So it's not spelling mistake. It is actually purposefully used that one. Okay. All right. Now, true, false questions. Glycolysis is a preparatory process for producing more ATP. Good. Next one. Glycolysis is activated by glucagon, insulin, cortisol, adrenaline. Glucagon inhibits glycolysis. Insulin activates glycolysis. So make sure, don't get mixed up with that. Okay. So the last one, glycolysis occurs in which part of the cell? Is it mitochondria? Is it nucleus, lysosome, or is it the cytosol? How come we have only 2 people answered? Cytosol? And nucleus? No, it can't be nucleus. It's not in the mitochondria, it's in the lysosome. So every metabolic process that you discuss in your assignment, you need to include this as well, where it happens in the cell. Okay. So that finishes, these 10 questions are actually the key concepts based question. Okay. So that's what you have to take forward from this particular session. Okay. Any questions so far?
Is this going to be recorded so we can review it?
Yeah. Of course,
Okay.
it is recorded. It is recorded. So that was fun. Thank you. Yeah. So this way, we are making sure that we know. Yeah, someone got a yeah. Okay. So now we move on to the next part. Okay. Can we move on to the next part?
Yes, thank you.
I was
So,
just wondering,
yeah.
is there any way to do the other pathways before
Yes.
Kahoot?
Yeah. I'm
Okay.
going to go through all the pathways like that. I have everything.
Oh, yeah. Before, because I was going to do protein metabolism. So
Okay. Yeah. You want today? I
Is
can
it- but
actually do it. I
is
can
there-
send you I can do send you a challenge number.
Oh,
Okay?
yes, please. That would be great.
Yeah.
Thank you.
So what I will do is I'll put a challenge number and post it to you, email to you. Okay. You send me an email. Yes. I need a Kahoot challenge quiz for protein metabolism. Yeah, I can do that. Okay. Is it who is it? Melissa or hello?
No, it wasn't you.
That was Kyla. Sorry.
Kyla. So you send me an email and I will send you the code for this challenge quiz. It is known as a challenge quiz. I can set it up as a challenge quiz. Okay. All right. So now what I'm going to do is I'm going to share this one. Session 19. You are able to see that? Session 19?
Yes, thanks.
Yes,
Common metabolic pathway. So why it is known as common? Because it is common for all the nutrients. Yes. So this is all the nutrients, your carbohydrate, lipids, and protein. When they are catabolized, the product is going to enter into the common metabolic pathway. So what is the product? 2-carbon compound. It is acetyl coenzyme A. Acetyl coenzyme A comes from, we know from pyruvate. Pyruvate comes from glucose, correct? Acetyl coenzyme is coming from glucose to pyruvate to acetyl CoA, right? That way. And acetyl coenzyme A, we can also get it from fatty acid oxidation. Acetyl coenzyme A also come from amino acid catabolism. The carbon skeleton of many amino acids produces acetyl coenzyme A. So that is also an input, right? And also glucose. So that's why CAC, citric acid cycle, is known as a part of the common metabolic pathway. CAC, citric acid cycle. Citric acid cycle is known by different names, right? What are the other names for citric acid cycle?
Krebs cycle.
Krebs cycle. Any other name?
Mm,
Tricarboxylic acid cycle. Citric acid is nothing but, with the 3 carboxyl group. That's why it's also known as tricarboxylic acid cycle. So CAC, Krebs cycle, TCA, these are all, this means the sign. Okay, different names. ETC stands for electron transport chain and OP stands for oxidative phosphorylation. Okay. Input for citric acid cycle. Input for citric acid cycle is acetyl coenzyme A. Yes. What else is needed? Oxaloacetate. And it also requires NAD plus and FAD also needs that. Okay. What is the output? NADH, FADH2 and GTP. So the electron poor, this is electron poor, becomes electron rich. Okay. So it actually gains hydrogen. Hydrogen is nothing but an electron and a proton. So NADH has hydrogen in it. So it is the reduced form of the coenzyme. If it has hydrogen, that means it has electron in it. So this is going to lose its electron. Loss of electron, gain of electron, right? So this has gained hydrogen, gain of hydrogen gets reduced, gets oxidized. Okay. So it loses hydrogen and proton. Yes. So the electrons are going to go through the electron transport chain. When electrons go through the carriers, at specific points, protons are pumped into the intermembrane space. Okay. Complex 1, complex 3, and 4, that is the proton pumping sites. So protons accumulate in the intermembrane space. They all move down gradient through ATP synthase. Energy from this proton gradient is used for making ATP, and that process is oxidative phosphorylation. So ETC and OP are concurrently happening processes. Okay. Important reactions of citric acid cycle: 3, 4, 5, 3, 4, 5, and 6 and 8. These are, we'll go through that, important reactions. So you have 4 redox reaction and 1 substrate level phosphorylation reaction happening in citric acid cycle. So then we will look at glycogenesis, making glycogen from glucose. Input is glucose, output is glycogen. Important reaction is, key reaction is glycogen synthase catalyzed reaction, whereby, you know, glycogen is made from glucose. Insulin activates this enzyme, and hormonal control: insulin activates, glucagon inhibits. And the importance of glycogenesis is storing excess glucose in the liver and skeletal muscle. But we have a limit to which we can store glycogen in the liver and muscle. If you are storing too much of glycogen in the liver, that is a disease. Okay, so it can lead to hepatomegaly. The liver enlarges. That's a disease actually. Glycogen storage disease it is known as. So we have a limit to which we can store. After that, excess glucose will be converted to fat by a process known as lipogenesis. Glycogenolysis is breaking down glycogen, and this is activated by glucagon and glycogen phosphorylase enzyme. Glycogen becomes glucose 1-phosphate, which becomes glucose 6-phosphate, and then glucose 6-phosphate is converted to glucose in the liver. In the muscle, glucose 6-phosphate goes into glycolysis because muscle lacks glucose 6-phosphatase enzyme. Okay. All right, so that's the concept map for this session. Okay, so the input for citric acid cycle is acetyl coenzyme. I have shown here fatty acid oxidation, amino acid catabolism, glucose catabolism, glycolysis, producing pyruvate. Under aerobic condition, it becomes acetyl coenzyme. This is all happening in the mitochondrial matrix. That's why I have put it in a box. Why it happens in the matrix? Anyone has any idea? Hello?
I know if I read it.
Because the enzymes are present in the matrix. Yes? If you know the metabolic process happens where the enzymes are present, what is the importance of enzyme? It reduces the activation energy, right? Speeds up the reaction rate. All the enzymes needed for citric acid cycle are present in the mitochondrial matrix. Pyruvate acetyl coenzyme, the pyruvate dehydrogenase enzyme, which is also present in the matrix. Okay. Now that's the citric acid cycle. I have represented citric acid cycle. The important steps I have shown there. Step 1, that's where citrate is formed. This step is important because in the fed state, when we have made enough ATP, citrate will be taken out of the matrix into the cytosol using a carrier there, malate aspartate shuttle, citrate malate shuttle. So I'll talk about that later. So that's why I have shown this step. Okay, 2 carbon, 4 carbon compound becoming 6 carbon. So oxaloacetate is the last step of citric acid cycle, which is used for the first step of the citric acid cycle. There, citrate, isocitrate, important step here. 3 is important, NAD is used. 4 is important, NAD is used. See, carbon dioxide is lost. 2 carbons are lost like this. Acetyl coenzyme is a 2 carbon compound. Both the carbons are lost there. Okay. Then succinyl coenzyme to succinate. So that is substrate level phosphorylation. GDP is the substrate. GTP is the product. Yes. And then succinate to fumarate. That's a redox reaction. FAD becomes FADH2. So 3, 4, 6 and 8 are redox reactions. And step 5 is a substrate level phosphorylation reaction. So that's the key steps which you need to remember. Okay. Output 3 NADH, 1 FADH2 and 1 GTP, which is equivalent to 1 ATP. Okay. Oxaloacetate is important for citric acid cycle. If not in oxaloacetate is there, then citric acid cycle will not function. Oxaloacetate comes from pyruvate by pyruvate carboxylase enzyme. So if glucose doesn't enter into the cell, oxaloacetate will not be made from pyruvate. Okay. So that becomes a limiting factor. So citric acid cycle will not proceed without oxaloacetate. So diabetic patients, untreated diabetic patients, what is going to happen? Citric acid cycle is going to be affected because glycolysis is going to be affected. Yes? So where the acetyl coenzyme can go? To ketone bodies. So that's the lipid lecture, lipid metabolism 2. All right, glucose in the fed state, when you have excess glucose, see, by a process of glycogenesis, glucose 6-phosphate becomes glucose 1-phosphate. Glucose 1-phosphate combines with the UTP to form UDP glucose, glycogen synthase. This enzyme is activated by insulin. That's why you have glycogenesis happening in presence of insulin. Glycogenesis starts with the glucose, ends with the glycogen, activated by insulin, inhibited by glucagon. So glucagon by a second messenger system is going to deactivate glycogen synthase, but glucagon is going to activate glycogen phosphorylase. Glycogen to glucose 1-phosphate, glycogen phosphorylase. This enzyme is activated by glucagon, inhibited by insulin, and glucose 6-phosphate to glucose. This enzyme, glucose 6-phosphatase enzyme, is present only in the liver, not in the muscle. Okay. The role of insulin and glucagon shown there, right? Blood glucose level, when the blood glucose level is high, insulin is released from the pancreas. Insulin stimulates glucose uptake. Insulin promotes, stimulates glycogen formation. Okay. When there is low blood glucose level, you need to increase the blood glucose level. So glycogen is converted to glucose. Yes. And also glucagon is responsible for gluconeogenesis as well. So these two counter-regulative hormones you have to remember. Okay, any question from this slide?
Is it just memorizing and understanding the whole
Understand.
process?
So when you draw like this, you will understand. Yes.
Yeah.
You need to scribble down. Don't simply look at that. Okay.
Yeah.
Yes. So if you have a whiteboard, it is good to draw the, what you take it glucose to glycogen and back to. So if you draw few times, you will remember. It's so important later on, because if someone lacks a glucose 6-phosphatase enzyme, what's going to happen? Any thoughts? They will be presenting with hypoglycemia, because between meals or when you are fasting, your blood glucose level has to be brought back to normal, correct? If that doesn't happen, there is something wrong, an enzyme defect. What about if someone has glycogen phosphorylase defect? Again, same problem. Yes, glycogen phosphorylase is present in the liver and muscle. So if the liver glycogen phosphorylase is defective or lacking, they will present with the severe hypoglycemia. Or if the muscle glycogen phosphorylase is defective, what is going to happen? They won't present with the hypoglycemia. Instead, they will not be able to do exercise. Because when you are exercising, the glycogen which is present in the skeletal muscle is used for making glucose 6-phosphate. See, a lot of relevance is there, these metabolic process that you are looking at, right? So in clinical medicine, we look at, what goes wrong in patients with hypoglycemia, okay? Or inability to do exercise. Are you
I've
okay with
heard
that?
that there is a technique that the athletes use when they're running marathons and things.
Oh yeah, of course. They use glucose loading and so on, carbohydrate loading and so on.
Yeah, but it's like a technique that they don't use the glycogen until they actually.
If you are a marathon runner, it is, your glucose will not give enough ATP for them, correct?
So what do they do?
They use fatty acid.
Oh,
Fat.
yeah.
You can see all the marathon runners, the Ethiopian runners and so on. You might have seen how skinny they are, right?
Yeah. How do
Because
they do it?
yeah, so they need heaps of ATP, which glucose can't give. Glucose gives only about 30 moles of ATP, or one glucose molecule can give only 30 ATP. Whereas your fatty acid depends on what fatty acid is. If it is a C12 or C16 fatty acid, you get 100 ATP also. See, look at the number of ATP you get from fatty acid, far more. Actually, 2.5 more, times more moles of ATP you get from fatty acid.
So how do you isolate the glucogen and start using the fatty acids instead?
No, they use initially glycogen, these guys, okay?
Ah.
Yes, but your glycogen will be finished in no time, because how much glycogen you are storing in this muscle?
Right,
It's about,
I understand now. Yeah.
yeah, it's
Yes.
about only 300 grams. That's all.
All right.
Yes, about 150 grams we are going to store in the liver, about 300 in the muscle. That's all.
Oh, gotcha.
See, that's why after 2 hours or so, your glucose level is going down, and then we are using
Fatty acids.
glycogen. We use glycogen. If you are continuing fasting, then you need to get the glucose from non-carbohydrate sources. That's what happens. That's a process known as gluconeogenesis.
Okay. Thank you.
Yes, done this one. Any question there? Now let's go through this one quickly, and then I will do the Kahoot question. Okay. What reactants are required for citric acid cycle? Include coenzyme.
Acetyl-CoA and the
Yes.
oxalase.
Low-acetate.
I can't remember the name. Yeah.
What is the coenzyme needed? NAD and FAD. FAD.
Yeah. Okay.
Yes. So here comes the answer. Acetyl coenzyme, oxaloacetate, NAD and FAD. Okay. What happens to the coenzyme NAD plus FAD in the redox reactions of CAC? How does this affect the reactant compound that the coenzyme react with? NAD FAD becomes?
Oxidized.
Yes. Oxidized or reduced?
Oh, no, they're reduced. No, they've been oxidized.
No,
they've been
gaining
reduced.
hydrogen is?
They
Gaining
get
hydrogen, yeah, it becomes NADH and FADH2, which means gain of hydrogen, right?
Yeah.
Yes,
Reduced.
so that is reduction. So where the hydrogen has come from? The reactants, right? So reactants loses the hydrogen, the coenzyme gains the hydrogen. So reactant gets oxidized and the coenzyme get reduced, correct? Coenzymes gets reduced, reactant gets oxidized. How does the creation of electron-rich coenzyme NADH, FADH to link CAC to ETC? Any thoughts? This is the one which is going to give electron and proton, correct? Electrons are going through carriers, yes? And protons are going to go through this way, Yes, complex 1, complex 3, complex 4. Yes, nothing happens in complex 2. Okay, so the flow of electron is like that, and then it goes into oxygen. Oxygen is the final acceptor of electrons from the electron transport chain. The electron rich coenzyme are taken to the ETC, released. Electrons from the NADH and FADH are released into the ETC. Allows a large amount of ATP to be made. That's the significance of oxidative phosphorylation. These two processes are happening concurrently. Okay. Through what metabolic process is glucose storage polymer glycogen made? Under what circumstances? Yes,
Glycogenesis,
correct. Under what circumstances?
excess glucose,
Yes. When are we going to have excess glucose?
or we've eaten too much.
Yes, good. That is known as fed state, right? Fed means you have eaten heaps of carbohydrate, right? So glycogenesis in the fed state when there is excess of glucose. Good, well done.
Okay. 'Cause I
Is
was thinking,
glucose, yes?
sorry, I was thinking like if you've actually eaten too much, but it's regardless. It's just if you've eaten, that process is going to happen, right?
Yes, it is
Yeah.
going to, because when you eat, you are going to eat carbohydrate a lot, correct?
Yeah, tend to.
Yes. Yes, you don't need all the glucose which goes into the cell to be made ATP, because remember ATP is the glycolysis is tightly controlled. Step 1, 3, and 10 are regulatory steps, correct? Why do we need these regulatory steps? If all the glucose we are going to convert it into ATP, we will be making a lot of ATP. We will be jumping up and down with ATP. Yes.
Yeah. Okay.
That's not going to be, help anyway our cell, right? When we have made enough ATP, or when the cells are charged with ATP, ATP itself is going to inhibit those regulatory steps, right?
Mm-hmm.
So that we can slow down glycolysis, we can activate glycogenesis.
Mm-hmm.
That's what happens. If
Cool.
glucose is in excess in blood, where excess glucose can be stored as glycogen? And for what purpose?
The liver and muscle.
Liver, and we have to be very specific which muscle, right?
Oh, it's the skeletal muscle, isn't
Yes, skeletal muscle.
Okay.
Because heart doesn't store glycogen.
it? Okay.
That's why. For what purpose? Why do we store? So
when
that
we need, when glucose levels are low, that we need.
when glucose levels are low, we can actually release glucose from glycogen. Between meals, when you are exercising, or if you are fasting, correct? That's when we need glucose, so that glucose dependent organs can get it, correct? Liver and muscle, and it can be broken down into glucose to produce energy. I took straight from the notes, but I'm not happy with saying simply muscle. I want it as a skeletal muscle. Okay.
Yeah.
Yes. I purposefully included like that so that I wanted to highlight that muscle we have to say it is skeletal muscle. Okay.
Okay.
What metabolic process is used to break down glycogen?
Glycogenolysis. How can I pronounce it?
Glycogenolysis. We need to familiarize with these words, right? Terminologies. Glycolysis, glycogenesis, glycogenolysis, and gluconeogenesis. They get confused, okay? Which is which you have to familiarize. And glucagon and glycogen are not the same. Glycogen is a polymer, glucagon is a hormone, okay? Glycogenolysis to produce glucose so that organs that does not have glucose reserve. Which organ does not have glucose reserve? Anyone? Hello? Hi.
I'm not sure. What was the question again? Sorry. Which organ?
Don't have glucose reserve. Brain. Brain does not have glucose reserve. Brain doesn't store glucose. Brain doesn't convert glucose to glycogen. Brain doesn't store fat. Yes? And brain also does not store proteins as well. If that happens, that is a disease, right?
Okay.
Lipid storage disease, very dangerous disease which can damage the brain. Yes, it's a lysosomal enzyme defect. If proteins, condemned proteins accumulate, that's also a disease, like the Alzheimer's disease, beta amyloid protein accumulation, damaging the neurons and so on. See, brain don't have any energy reserve. So we have to constantly provide glucose. The major consumer of glucose is brain. The moment glucose is low, what is a symptom of hypoglycemia? You get disoriented, right? Okay. Now we will go to the next Kahoot question. Yes. Any questions so far? Hello.
No, thank you. Not at the moment.
Not at the moment. So we will go back to the next set of Kahoot questions, right? And I need to generate my Kahoot. Carbohydrate metabolism 2. Let's see. Where is carbohydrate metabolism 2? So teach. All right. Are you there? Can you see the new pin?
Yes, thanks.
Yeah. We have one more person to join. Start. Okay. So the citric acid cycle occurs in the is it cytosol, nucleus, mitochondrial membrane, or the mitochondrial matrix?
Mitochondrial matrix.
Okay, matrix is the one. That's where the enzymes for citric acid cycle are present, not in the mitochondrial membrane. So in the mitochondrial inner membrane, we have the which process happening in the inner membrane? The electron transport chain. In the nucleus, we have protein, DNA replication and transcription, and translation is happening in the cytosol. In the cytosol, we have glycolysis, glycogenesis, glycogenolysis, lipogenesis. All those things are happening in the cytosol. Okay? Cellular location is very important for us to know. Okay, the common intermediate Acetyl coenzyme that enters the citric acid cycle is coming from glucose, amino acids, and fatty acids. Okay, all your macronutrients are used for or for when they are broken down becomes acetyl coenzyme, which feeds into citric acid cycle. All right, how many NADHs and NADHs are produced in the CAC? Three NADH, right? One FADH2. How many GTPs are produced? Remember the substrate level phosphorylation, succinyl coenzyme A becoming succinate. So that step. One, good. CAC is allosterically regulated at step. Which step? When we have high ATP or high NADH, when we have more NADH means you are going to make more ATP by electron transport chain. So that's going to inhibit citric acid cycle. Step 3 and 4. NADH, FADH2 are source of electrons and proton, oxygen, carbon, and water. Good. That was very quick. Yes. I'm happy. Which one of the following is the electron-rich coenzyme? NAD+, NADP, FAD, or NADH? NADH. NAD plus is the electron poor, right? Because it does not have hydrogen associated with that. Hydrogen is nothing but electron and proton. One electron, one proton. One turn of CAC produces how many ATPs when combined with ETC and OP? So this is actually coming from the next one, because we haven't done that in the CAC. Sorry for that, right? So we are going to produce 3 NADH, 1 FADH2. So which means each NADH is going to give us 2.5. So 3 times 2.5, 1 times 1.5 plus 1. So that is how it becomes 10. So I will do that calculation in the next session. Sorry for that. How many ATPs are produced from, not from it should be, sorry for that. Again, this is related to the next one. Okay, I said a while ago the number of ATP from NADH and FADH2. Good. One you always have to remember one NADH is 2.5, one FADH2 is 1.5. So that's the one which we are going to use for the number of ATP produced from CAC or from fatty acid oxidation. The basis for calculation is comes from here. Okay. True or false? FADH2 produces 1.5 ATP from ETC and OP. Good. Nice. Synthesis of glycogen from glucose is called glycolysis, glycogenolysis, glycogenesis, gluconeogenesis. Good. Glycogenesis. Glycogenolysis is the breakdown of glycogen from glucose. Good. Glycogen is stored in the liver, brain, heart, and kidney. Good. Glycogenesis is activated by glucagon and inhibited by insulin. True or false? It is false. Glycogenesis is activated by insulin and inhibited by glucagon, the other way around. Okay. Yay, Kyla, well done. All well done. Good. Okay, so now question time.
I actually have a couple of questions, but this is in relation to session.
Give me a few seconds. I will share a different screen.
But it's in relation to session 20, so I'm not sure if you.
No, I can answer that. No problem.
Okay, cool thing.
Oh,
Yeah.
sorry, are we doing session 20 today or?
No, now we are up to time is so finished time.
No worries, thank you.
Yeah. So we don't have enough time to do that. I can post the concept map 19 and 20. Yes.
That'd be great, thanks.
Yes, I will post that. Yes. Question from 20.
So the first one is actually on the slides when it's going through the electron transport chain. So in complex 1,
Mm-hmm.
on the slides, the lecture slides, it says that NADH loses 2 H plus and 2 electrons.
Yes, it is picking up actually from the cytosol, from the matrix actually. Energy
Yeah,
don't
That's
have.
what I get confused with, because in the
Yeah,
next, then the
that
next
much
slide,
detail. That much detail. Yeah, when you add up. So this is the question we always get if you carefully look where the other hydrogen is coming from.
yeah, but it says,
Yes.
on the first slide it says it comes from NADH, but then on the next slide it says that one of the H
It's actually coming from the matrix, so it is going to pick up and goes like that. So
Okay.
in our textbook does not have any explanation. If you read which one is it? Is Stryer textbook by Stryer. So he explains that in detail how the other hydrogen comes. That is too complicated. That's why I don't think we need to worry much. If I manage to. Yeah.
Okay, cool. So just to clarify, so with NADH, that just has 1 hydrogen and 1 electron. Sorry,
It
one
has one
proton.
hydrogen there. It is 1 electron and 1 proton.
Yeah. So then another electron and
It is
proton
coming
is
from. See,
elsewhere.
remember we always say NADH plus H plus. Remember, so that's the one which is it's the one that's it's picking up.
So sorry, so
NAD plus becomes NADH plus H plus.
Oh, because it doesn't actually sort of in the text, it just says NADH.
No, there is a, in your reaction, which is reaction is that 3 and 4, 3, 4 and which one? 3, 4 and 7. 3, 4 and 7. What we are producing is NADH plus H plus. Yes? It is not shown there probably.
Yeah, no, it doesn't show the
Yeah.
plus. That's where I'm getting confused, I think. Yeah, okay.
I understand now. Yeah. So it is actually picking up while going there. So I can actually post it to some, if you need extra information on that, how another H goes in there. Okay. I can do that. Because
Yeah,
I don't want to confuse people more, right?
no, yeah, okay. It does say that it gets the H plus from the cellular solution, but
Yeah, yes, that H plus is actually, see, all those reactions, 3, 4, 7, and in fatty acid oxidation reaction as well, you are going to produce NADH plus H plus. Whereas
okay.
FADH2, it is H2. There are 2 hydrogen there. There is no issue there. Yeah.
How come it doesn't say NADH plus though?
NADH plus H plus, it does. It is, they sometimes these books thinks that what it is.
Oh, okay, okay.
Yeah.
Thank you. And the other question is actually number 7 in the tutorial.
Mm-hmm.
It gives
Yep.
it gives the equation and asking what's missing, and it says that there's half an oxygen.
Oh, okay.
Oh, yes,
O2,
thank you.
O2 plus 4H gives you 2H2. Okay, 2H2O. Now, is it half O2? Is it half O2 it says?
Yeah, it says half O2 plus 2H plus question mark equals H2O.
H2O. Because you can't have half O2, you need to multiply the whole equation by 2. Yeah. So that's how you end up with O2 plus 4H plus gives you 2H2O.
Why are we multiplying it by 2?
Because you can't have a coefficient in fractions. Can we? There is a coefficient of 1 in front of O2, right? Yes, no?
Yes.
Yeah, you can't have an equation in half. So you need to double it up. So when you double it up, it becomes O2 plus 4H plus 2H2O. It is eventually going to happen like that. You don't take half O2, instead you take only O2. The textbook initially says it is half O2 plus 2H plus gives you H2O.
Yeah.
But then they correct it saying that it is O2 plus 4H plus 2H2O.
Oh, sorry, I was saying yes to the question, but yeah, okay.
Yeah, the question, it is a balanced equation, but then the problem is you can't have a fraction in an equation. So that's, the rule is wrong, if you do like that.
So is it that there's two H2O, sorry, produced at the end? Is that?
Yes, of course, 2H2O, that is a cellular water. You have lots of protons coming there, correct? You have a gradient of protons coming there. So you have enough H plus there to make it 2H2O.
Oh, sorry. So the last step in the ETC, so we get 2 H2O. Is that what?
Yeah, you get to 2H2O.
Okay.
Yes.
Okay.
Because that's not correct in terms of coefficient that you are using. That's why.
So what is the actual correct answer for that, for question 7? Is it one oxygen, or is it 2?
No, if it says half O2 plus 2H plus is correct, but the problem is when you balance, you need to multiply that.
Mm-hmm.
Yes. So it is still correct whatever you have chosen. What is missing there? 2H plus, correct? I can't see the question.
Well, the answers give the answers that the options is ATP synthase, 2 electrons, 1 electron, or 1 oxygen.
Okay, so what is the correct answer it says?
I'm told it's 2 electrons is the correct answer.
Plus electrons, so I forgot to add the 2 electron there, so it will be 4 electron. Yes, if you balance out. So 2H plus and 2 electron becomes hydrogen, right? 2 hydrogen, correct? Yes, no? 2 hydrogen is nothing but 2 protons and 2 electrons, so it should be 2 electron.
Okay.
Yeah, whereas I initially went for 1, oh no, I think I went for 1, yeah, 1 oxygen, I think. But
Yeah,
yeah,
this is the equation that we use, and then we change it to a full equation because of a fraction we are using there.
mhm,
Yeah.
okay. That's a confusing question.
Yeah, it's 2 electrons.
Yeah,
2 electrons and 2 protons becomes H2. 1 hydrogen is 1 electron and 1 proton. So 2 means 2 hydrogen, which is H2, will have 2 protons and 2 electrons, correct?
yeah,
Okay. Any other question?
that was it, thanks.
Okay. That's done. Any other question so far?